This next lecture is going to be on nervous tissue. So putting this in perspective, remember there are 4 types of tissue in the human body. We have epithelial, which we've covered in detail, connective, which we've also covered in detail, muscle, which we just finished covering, and now we're finally on to nervous tissue. So functions of the nervous system in general is to receive and collect information, process that information, and then respond to that information. And remember, the nervous system works by sending electrical signals. So if we look at the organization of the nervous system, it can be divided into a central and a peripheral nervous system. So the central nervous system is your brain and spinal cord, and that is it, nothing else. The peripheral nervous system is everything else. So this consists of nerves, which are extensions coming off of the brain or spinal cord, and any other type of nervous tissue that's in the body. But if it's not within the brain or spinal cord, it is considered the peripheral nervous system. So if we look at a picture of this, we see the brain, we see the spinal cord, and so that's your central nervous system. And then all these nerves coming off, these are would be in the peripheral nervous system. So you get spinal nerves, which are coming off the spinal cord. And you also get some cranial nerves coming off of the brain. So a lot of students get confused when they see cranial nerves. They think, oh, that's the brain, it has to be central nervous system, but it's not. So if it's cranial nerves, it's outside of the brain. All right, functionally, it's a little bit different, but we can classify or organize the nervous system as to the direction that the electrical signals are going. So if the electrical signals are being transmitted towards the central nervous system, so that's towards the brain or spinal cord, from the peripheral parts of the body, we call that sensory. It's also known as afferent. And so I have some sub-bullet points under here. This is not overly important on the sensory side. It will be on the motor side, though. So difference, sensory information coming from skin, joints, and muscles is called somatic. If it's from internal organs, we call it visceral, but again, not a big deal here. So the other classification here is if the electricity is moving away from the central nervous system, so towards the peripheral nervous system or towards the peripheral parts of the body from the brain or spinal cord, we call that motor or efferent. So if you remember when we were talking about muscle contraction, we called the neuron that stimulated a muscle fiber, we called it a motor neuron because it was leaving the spinal cord and going towards skeletal muscle, which is in the periphery of the body. So if the motor nervous signal is going to skeletal muscle, we classify it as somatic, and that's what you've seen before. If it goes anywhere else, we call it autonomic. So that any place else could be smooth muscle, cardiac muscle, or glands. So here's a picture kind of depicting the different organization of the nervous system. So sensory versus motor. And then within motor, we have somatic versus autonomic. All right, so this you've seen before. The major cells that do the work in the nervous system are called neurons. So they respond to stimuli and send electrical impulses called action potentials. So these are excitable cells. So what you've learned before is that there's 3 major parts to a neuron. There's the cell body, which is called the soma. There's usually a bunch of extensions, depending on the type of neuron you're talking about, but this is the most common type. So you have a bunch of these smaller extensions called dendrites. And the dendrites receive information. Then there's usually one long axon that sends out the electrical signals. So the action potentials that we're talking about that are sent are transmitted down this axon. So if we take a closer look now, and we can go into a little more detail here. So here's that same picture. So I'm going to show you a few more anatomical features that we haven't seen before. So where the axon meets the cell body, this is called the axon hillock. This is important because this is where an action potential starts. So an action potential starts right here and it goes out towards another cell. So that's the electrical signal. It starts there at the axon hillock. Notice that this axon is branching. So axons branch, and each branch is called an axon collateral. Then as we get to the end of those axon collateral, you end up with these tiny little branches called axon terminals. They have them labeled as telodendria here, but axon terminals is fine for this. At the end of those axon terminals, there's these little round structures that look like little knobs, and they're called synaptic knobs. So the synaptic knobs are where the neurotransmitter is stored. So neurotransmitters are just a chemical that neurons use to communicate. So basically what's going to happen is, and you'll see this in more detail, the action potential or electrical signal is going to come down this axon, and when it gets to these synaptic knobs, it's going to stimulate the release of that chemical, that neurotransmitter. And we'll talk about that whole process as we go through this. Other features to be aware of. Notice that the axon in this picture is covered with something, and that white stuff is called the myelin sheath. So myelin sheath, that's an insulation layer around axons. It's not found on dendrites or the cell body. It's only on axons, and it's not every neuron that has this. So only certain neurons have this myelin sheath, and we call them myelinated neurons. Notice that the myelin sheath is not continuous. There's these gaps. So there's little gaps in the myelin sheath. Those are called nodes of Ranvier, and we'll talk about the relevance of that later in this discussion. Then lastly, I just wanted to point out that there's a specialized type of rough endoplasmic reticulum within neurons. It's called Nissl bodies, and that would be it right here. So remember, the rough ER is used for protein synthesis because it has ribosomes. All right, so here's a picture you've seen before. You got a neuron, axon, dendrites, cell body. All right, so the other cells of the nervous system, other than neurons, remember there's these other cells called neuroglial cells. These are thought to be supporting cells. They outnumber neurons, so there's a lot more of these neuroglial cells than there are neurons. They can divide, which is something that neurons can't do, which is why it's so bad when you get damage to nervous tissue. These are smaller than neurons, but there are different types. There's 6 different types of these neuroglial cells. And again, they don't generate action potentials. That's what it says up here. So they don't really do the hard work. They play a supporting role, and each of the 6 has a specific role that it plays in supporting the nervous system. So notice that 4 of these, it says CNS behind it. So these first 4 are found within the central nervous system, and these 2 are found in the peripheral nervous system. We're going to go through these one by one, and I'll tell you what you need to know about each. So starting with astrocytes. So astrocytes are in the central nervous system. They get their name because they kind of look like a star, and what they do is they form the blood-brain barrier. So if you look at what they're doing, they have these little feet. They reach out and they kind of wrap around blood vessels in the brain, and they prevent blood from mixing with brain tissue because that could be problematic. So they form this barrier called the blood-brain barrier. Over here we have ependymal cells. So ependymal cells line some spaces in the brain called ventricles, and they also line the central canal of the spinal cord. So those terms may be foreign to you right now, and that's fine. But just at this point, just understand that the brain has little cavities in it called ventricles, and these ependymal cells line them. And the spinal cord has a canal that runs right down the middle called the central canal, and that's also lined with these ependymal cells. So when all these ependymal cells get together, they form a structure called the choroid plexus. So all these ependymal cells together form the choroid plexus. The choroid plexus is important because it produces something called cerebrospinal fluid. That's the fluid that flows through your central nervous system. So ependymal cells produce the cerebrospinal fluid, which flows through the central nervous system. Notice that they have cilia also. That allows them to circulate the fluid. All right, moving on, microglial cells. Not a whole lot to say here. These are just phagocytic cells that eat and get rid of dead and damaged nervous tissue. Then we have one that's a little more interesting, oligodendrocytes. These are large cells. Notice that they have these little processes coming off of them, and their processes wrap around axons of neurons. So what they're doing is basically forming the myelin sheath. So the way these oligodendrocytes do it is they reach out with their little processes and they kind of wrap around different axons. So it looks like you can tell here that one of these oligodendrocytes can myelinate multiple axons. And the other thing I want to point out, just to be clear, this is within the central nervous system because these are central nervous system cells. So we're talking about myelination of neurons in the brain or spinal cord because it's going to be different for the peripheral nervous system. So moving on to the peripheral nervous system, we have satellite cells, which basically kind of form like a little barrier that goes around some of these. In this case, this is the sensory neuron, and we'll talk more about that later. But they regulate what goes in and out of the neuron. So satellite cells regulate what goes in and out. And then probably most important in the peripheral nervous system, we have these cells called Schwann cells. These wrap around the axon of neurons, forming the myelin sheath. So remember, now we're in the peripheral nervous system. So Schwann cells form the myelin sheath in the peripheral nervous system. They do so in a different manner than what we saw in the central nervous system. So each one of these little white things is a Schwann cell. So for instance, this axon is being myelinated by multiple Schwann cells. All right, and this is comparing the difference between CNS and PNS as far as myelination goes. So in the central nervous system, you have the oligodendrocytes. They're putting out their extensions, and they can myelinate multiple axons. In the peripheral nervous system, these Schwann cells have to devote basically their whole life to a part of one axon. And this is just showing you a Schwann cell wrapping around part of an axon. All right, so I wanted to give a brief introduction in how neurons work and talk about action potentials. And this is going to be a little bit of physiology, but I think it helps to understand what's going on, how an action potential works. So all cells, including neurons, have what's called a resting membrane potential. And what that means is that there's a charge difference, an electrical charge difference between the inside and the outside of the cell. So I drew a cell right here. Let's assume this is a neuron. So the inside has more negative charge, the outside has more positive charge. All right, so when you get that in a cell, we say this cell is polarized. So this is a polarized cell. If it was non-polarized, the charges would be equal across. All right, so there's a charge difference here, so we'll say that this cell is polarized. In neurons at rest, if the neuron is just resting, that charge difference is 70 millivolts. And we denote that as a negative 70 millivolt resting membrane potential. And the negative means that there's more negativity on the inside. So I want you, before I show you anything else, I want you to just think about this. So if this is polarized, what might depolarized look like? So depolarization of this cell would basically entail the inside becoming more positive, and so becoming less polarized. So that's a term that you're going to hear a lot in the next few minutes. Depolarization, basically that's the inside of the cell becoming more positive. And on the other term, another term you might hear, which we'll talk about later today, if the inside became even more negative, we could call that hyperpolarization, like excessively polarized. But let me move on and we'll talk more about action potentials. So neurons send electrical impulses called action potentials. We've established that. They start here at the axon hillock. They're going to go this direction. So action potentials occur when some stimulus causes the neuron to depolarize. So that's that inside becoming more positive. So what's going to happen is this. So if there's some stimulus, maybe some impulse or whatever altered this dendrite, so the dendrite received either electrical signal or something else, and it changed the inside charge of the cell. So if it became depolarized to a critical threshold, and that critical threshold is negative 55. If it gets to, let's see, remember it's at rest, it's negative 70. So if it got to like negative 60, nothing would happen. But if it gets to that critical level of negative 55, you're going to get a full-blown action potential taking off, going down this axon. And all an action potential is a wave of depolarization. And I'm going to try to explain that in the next few slides. So a wave of depolarization, and that's going to be followed by repolarization. So remember, depolarization would be the inside becomes more positive, and then repolarization is basically reversing that and going back to that resting state. So I want to show you this graph right here, and if this is too much to look at, you don't have to look at this, it's not imperative, but I do want you to focus on this graph. So this is a graph of voltage during an action potential. So let's just say we're measuring the voltage at a given point on an axon. And so at rest, that we said that resting membrane potential is negative 70 millivolts. So here we are, we're at negative 70. Then there's some stimulus. And that stimulus, let's say this first example, gets it to negative 65. Nothing's going to happen, right? Because it didn't hit that magic number of negative 55. Then maybe a bigger stimulus came along and got it to negative 60. Nothing's going to happen because it didn't get to that critical threshold. If even a bigger stimulus came across and got it to negative 55, then you get this very fast depolarization followed by a repolarization. And that is basically what's going to happen during an action potential. So once you get to that critical threshold, the quick depolarization, so notice that the voltage is going from negative 70 to negative 55 to past 0, even into the positive numbers. That means there's more positive charge on the inside. So this rapid depolarization is caused by an influx of sodium ions. So the sodium ions rush into the axon or into the neuron. And then after a very brief period of time, you're going to get repolarization, and that's due to potassium ions moving out of the cell. So that requires a little bit of thought. So both sodium and potassium have a positive charge. So sodium rushing into the cell is going to make the inside of the cell more positive, and that's the depolarization. In turn, potassium also has a positive charge. It's going to rapidly leave the cell. When positive charge leaves the cell, that brings the inside of the cell back to being more negative, and that's the repolarization. So key point, the depolarization is due to sodium ions rushing into the neuron. Repolarization is due to potassium ions flowing out of the neuron. So that's going to happen at each little section of the axon as it makes its way down the axon to the end. And so I have a lot of pictures here, so I'm just going to kind of show you what I'm getting at. So this is like a snapshot. So if this is part of the axon, at this point in time, that this little section of the axon is depolarized. Assuming that the action potential is moving this way, this has just been repolarized, and then this is still at rest. Now you can imagine what's going to happen at the next moment in time. This is going to be depolarized while this is repolarizing. And it's just like a wave that goes down the axon. So if I do this fast enough, it kind of looks animated, like boom, boom. And so that just shows you that it's like a wave of depolarization followed by repolarization. I always kind of relate this to if you were at a sporting event and people in the stadium started doing the wave going around the stadium. So the people standing would represent the depolarized area. The people that just sat down would be repolarized, and the people who haven't stood up yet would still be in the resting state. So with that being said, there's a period of time after an action potential where a neuron cannot generate another action potential. So we call this refractory periods. So it's kind of like recovering. So there's an absolute refractory period and a relative refractory period. During the absolute refractory period, there's just no possible way that an action potential can be generated. It's just like absolutely impossible to generate an action potential. And this is a very short period of time. But just after that, you could generate an action potential, but it requires a bigger than normal stimulus. And so that's called the relative refractory period. All right, so I want to show you this to get an idea of why this might make sense. So like maybe at this point in time during repolarization, it's impossible to generate another action potential. But right here, it looks like there's kind of this undershoot period. You could generate an action potential, but it's going to take a larger than normal stimulus to get to that negative 55 because you're actually starting at like negative 80. So it's not truly at rest yet. So this would be like in the area of a relative refractory period. Over here would be impossible. So this would be absolute refractory period. All right, so that's refractory periods. And then just more pictures for you. So kind of showing you the different phases as it's moving down the axon. And the refractory periods are important because this ensures that the action potential only moves in one direction. So in other words, if the action potential is moving this way, like at this point in time, this period is still in that absolute refractory period, so it's impossible for this action potential to turn around and go back this way. So it has to keep going in one direction. So you only get action potentials moving from the cell body towards the end of the axon. And so this kind of reinforces what we were saying. So depolarize, next section depolarizes while this is repolarizing, and then keeps going. So at this point, this is still in the refractory period. This is good to go for another action potential because it's back at rest. All right, how does this differ in myelinated versus unmyelinated axons? Well, the purpose of myelination, we called it insulation, but really what it does, it speeds up the action potential. So, to show this, I'm gonna start with an unmyelinated axon. That's what we've been talking about. So we call this action potential on an unmyelinated axon. We call it continuous conduction, meaning every little section of that axon has to be depolarized and repolarized. So if you compare that to a myelinated axon, where you have these, if you kind of look here, you have the big Schwann cells or oligodendrocytes, whichever nervous system you're in, they're kind of covering it up, right? So if we zoom in on that, it kind of looks like this. Here's a Schwann cell, for instance. So the sodium and potassium can't get through the myelin. So you can only get depolarization and repolarization at the nodes of Ranvier. And that's important because if you only have to depolarize at the nodes versus depolarizing every little section of the neuron, this is going to be much faster. So it's basically allowing the action potential to skip big chunks of the axon. And in turn, it's going to move a lot faster. So we call this saltatory conduction. Sometimes it's pronounced saltatory, but this is much faster than continuous conduction. So myelination can speed up action potentials like over 10 times. All right, the all or none law of action potentials basically says that an action potential, once generated, will travel all the way to the end of the axon and it never loses strength. So it's not like you can have a big action potential or a small action potential. They're all the same strength. And once you start it, you can't stop it until it gets to the end. So what happens at the axon terminal? So earlier I alluded to the fact that there's neurotransmitter released. And so when an action potential reaches the end of an axon, a neurotransmitter, which is a chemical, is released. There's two really common neurotransmitters in the body that we'll see this semester. Norepinephrine is one. Acetylcholine is the other. You've actually seen acetylcholine in the muscle lecture already. So that chemical or neurotransmitter is going to cross the space that when it's released into. It's called a synapse or synaptic cleft. And it's going to bind receptors on another cell. And that cell could be a muscle cell like you've seen before. Could be a gland, an endocrine gland. Could be another neuron. It could be any other cell of the body that the neuron is communicating with. So if you look at pictures, here comes the action potential. And this is the neurotransmitter being released. These are the receptors on the cell that's waiting. The space here would be called the synapse. So we could say this is the postsynaptic cell. And the neurotransmitter is released and it's communicating with the next cell. So notice the link between the action potential and the release of the neurotransmitter is calcium. So basically what's going to happen is that electricity, the ion, the electrical charge change coming down due to the action potential, is going to open up these calcium channels, which are responsive to voltage, and it's going to let in calcium, and calcium is going to cause the neurotransmitter to be released. And this is just more pictures kind of taking you through that process. All right, so oftentimes you do get one neuron stimulating another neuron. So the first neuron, if you have a setup like this, the first neuron is called a presynaptic neuron. The second one is called a postsynaptic neuron, because the synapse would be between the two, that little small space that you can't even see right there. So if you think about this presynaptic neuron could either stimulate this second neuron or it could inhibit it. And so if you wanted to stimulate this postsynaptic neuron, you would want the response here to be depolarization, to make it more likely that this neuron is going to get to that critical threshold of -55, because it's already at -70. If you want to stimulate it to send an action potential itself, you got to get it to -55. So it would make sense to have a depolarizing stimulus here. On the other hand, if you wanted to inhibit this neuron, you would want to get it as far away as you can from -55, right? So it's already at -70. So if you wanted to make sure that this second neuron did not send an action potential, you would hyperpolarize it, meaning take it from -70 to maybe -80. That makes it less likely that this postsynaptic neuron is going to generate an action potential. So I have some terminology about that. So the stimulus at the postsynaptic neuron can be excitatory or inhibitory. So if you want to excite that postsynaptic neuron, we call it an excitatory postsynaptic potential. And you would, like we said, depolarize the postsynaptic neuron. If we wanted to inhibit the postsynaptic neuron, we call that an inhibitory postsynaptic potential, and we would hyperpolarize the postsynaptic neuron, meaning take it further away from that -55 possibility. All right, just want to have a brief slide on convergence and divergence. So commonly what you'll see is neurons working together. And so in this is called convergence, where you get multiple neurons kind of all merging into one, stimulating one neuron. And then divergence is the opposite. So you have one neuron that stimulates many neurons. So oftentimes you see on sensory circuits, you'll have something like this, where you get information coming from multiple sources coming into maybe one or two neurons. And motor seems to be more like this, where you have one neuron going out, but it branches and communicates with many other neurons. So convergence, divergence. So some other terminology that I just want to throw out there. If you ever hear the term nerve fiber, which is not that common, they're talking about an axon. But the next four are important. A bundle of axons in the peripheral nervous system is called a nerve. A bundle of axons in the central nervous system is called a tract. So technically you have no nerves in the brain, right? You only have tracts in the brain and spinal cord. So a bundle of axons in the central nervous system is a tract. Bundle of axons in the peripheral nervous system is called a nerve. Then oftentimes these neurons communicate at places where you get a bunch of just cell bodies clustered together. So cluster of neuron cell bodies in the peripheral nervous system is called a ganglion. You'll see a lot of that when we talk about the autonomic nervous system. And then if you get that same thing in the central nervous system, it's called a nucleus. So this is different from the nucleus we've talked about of cells. So a cluster of cell bodies, neuron cell bodies in the central nervous system is called a nucleus. All right, just like muscle, nervous tissue has connective tissue coverings around it. And the terminology is really similar. So around an individual axon, so this would be on top of the myelin sheath if it's there, you would have connective tissue called the endoneurium. The axons bundle into bundles called fascicles, just like muscle did. And the connective tissue surrounding a fascicle is called the perineurium. And then around the entire nerve, we have the epineurium. So very similar setup to muscle, just the terminology is different. So picture, here's an axon with the myelin sheath, endoneurium. This is a fascicle, so it's going to be surrounded by perineurium. And then the entire nerve is surrounded by epineurium. All right, neurons can be classified structurally and functionally. So functionally is pretty easy, and we've actually done this already. So a neuron that transmits information towards the central nervous system, this is your spinal cord, and this is how it looks like in the skin. So this would be a sensory neuron, because it's carrying information towards the central nervous system. Motor, or efferent, carries information away from the central nervous system, maybe out to muscle in the body. So sensory towards the CNS, motor away from the CNS. Sometimes within the CNS, we have these neurons that kind of connect the two, and these are called interneurons. It literally means between neurons. They're also known as association neurons. So they're within the CNS, and they're kind of connecting the sensory and motor neurons. All right, structurally it's different. And the neuron that I've been showing you so far is called a multipolar neuron, and we always show that one because it's the most common. So I'll do that one first. So multipolar neuron is basically the neuron that we've been looking at. So it has one axon, several dendrites. Here's a good picture one. Cell body. So one axon, many dendrites. That's basically what we've been looking at. All of your motor neurons are multipolar, and interneurons are also multipolar. All right, then we have some differences. I'm going to go back up to unipolar. So unipolar looks like this, has a weird look. It has a cell body, has one extension coming off of it, and then two branches, and then both labeled as axons, and at one end you have some dendrites. So this is what sensory neurons look like. These are classified structurally as unipolar neurons. So sensory neurons, most sensory neurons are unipolar. They look like this. And then lastly, we have bipolar. Bipolar are really rare. So, but they're also sensory. They're used in special senses. So specifically vision and smell. So they look like this. They have a cell body on one side. They have a dendrite on the other side, an axon. And like I said, they're really rare. They're found in the retina of the eye for vision. Also found in olfactory neurons, which are used in smell. And then the last thing I want to finish up with is gray versus white matter in the central nervous system. So white matter is white because it has myelin. So we know that the only things that can be myelinated are axons. So the white matter is myelinated axons. The gray matter is everything else. It just appears darker. So gray matter, no myelin. That includes cell bodies or somas, dendrites, and also unmyelinated axons and neuroglial cells. So this, what you're looking at in this picture, is a cross-section of the spinal cord. So in the spinal cord, you have gray matter in the middle, white matter on the outside. And in the brain, it's a little bit different. You get gray matter on the outside, white matter in the middle, and then very deep in the middle, you get some more gray. So in the brain, it's gray, white, gray. In the spinal cord, outside to end, you get white, then gray.